All right, I'm going to go ahead and get us started because it is 12 o'clock on the dot. I am on service. I apologize. I feel underdressed for grand rounds today. But I want to get us started with an introduction. Just as a reminder, Medical Grand Rounds is back in person. We're going to continue to broadcast on Zoom for those who can't make it, but we want everyone to come here in the Bruce Stroffel Auditorium, fill this up every week as possible, please. As a teaser to making that happen on November 8th, I am really excited that we're going to present the 2023 Tim Gill Distinguished Lectureship that will be given by Dr. Dan Baroque, who's the director of the Center for Virology and Vaccine Research at Beth Israel and Harvard Medical School. And then on November 15th, we're going to welcome our own Dr. Joyce Lee, who's going to talk about complications and the impact of autoimmune lung diseases. And as a reminder, there is CME and MOC credit for all grand rounds this year. Before I introduce our speaker, Dr. Anne-Marie Schmidt, who I am very excited to tell you about today, I want to talk about the lectureship itself. So today is the 2023 Memorial Robert H. Eckel Lectureship, and we really are happy. And Bob is well, it is in your honor for sure. And of course, Bob is here, and we're really happy to welcome him, his family. And to put this on again this year, and this is a lectureship that's gone on for a number of years now. Bob is, suffice to say, a legend around here. He is a professor of physiology and biophysics, the Charles Betcher Endowed Chair in Atherosclerosis within our Department of Medicine. His translational research really led and defined the field and continues to do so as it has for the past several decades. He is the past president of the American Heart Association, the Obesity Society, the Association of Patient-Oriented Research. He co-chaired the 2013 AACC-AHA Lifestyle Guidelines, which many of you are familiar with and have based a lot of your care on, again, for a number of decades. So it really is an honor for me to be able to speak about Bob, to see him here today, and to say thank you for making this lectureship possible. Okay, now I am pleased to welcome today's speaker, Dr. Anne-Marie Schmidt. Dr. Schmidt is the Ivan Young Professor of Endocrinology in the NYU Department of Medicine. She also holds professorships in the Department of Biochemistry, Molecular Pharmacology, and Pathology, also at the NYU Langone Medical Center. She's the director of the Diabetes Research Program at the NYU School of Medicine, and she is the current editor-in-chief of the journal Atherosclerosis, Thrombosis, and Vascular Biology. I always go through everyone's academic history. Anne-Marie's is pretty concise. She went to NYU for undergrad, where she graduated summa cum laude in history and biology, for medical school, for her internship and residency, where she was a chief medical resident. She then did fellowships in both hematology and medical oncology at NYU. And she was a postdoctoral research fellow at Columbia University, so down the road a little ways, in the lab of Dr. David Stern. Dr. Schmidt has also been a leader and really is an exemplar of someone who can speak to and represent Bob Eckel and his work in the treatment of, in the field of biology for diabetes, obesity complications for decades now herself. She has over 425 original manuscripts and articles, as well as chapters published on a wide range of topics. Her identification and discovery of the receptor for advanced glycation end products, which we'll hear about today, was the result of decades of work both at Columbia and at NYU. It's had an incredible impact in the field of diabetes and cardiology and endocrinology, obviously, but also beyond that, extending into the fields of Alzheimer's research, rheumatoid arthritis, ALS, and a host of other diseases. Truly is my pleasure to welcome Dr. Anne-Marie Schmidt to Medical Grand Rounds today.
Well, thank you so much. That's very generous. Yeah. Sometimes I hear those things and I look around like, where they talking? Yeah, but thank you. Thank you very much. We all know how hard it is day to day to keep ourselves going and move forward. So I appreciate the synthesis. Thank you. So today I'm very honored. You know, I had a wonderful meeting with Bob and Margaret, his wife, and I have to say that it is very true that so many of the things that you just said about the work that we have done really relies on the discoveries of others who have contributed so, immensely to these fields. So today I'll tell you the RAGE story, where we are and where we hope to be able to move this, hopefully in the near future, into the clinic. So the RAGE was discovered by virtue of its ability to interact with advanced glycation end products. So AGEs are the products of non-enzymatic glycation and oxidation of proteins and lipids. And basically, it's a gain of function that when these modifications form irreversibly on proteins, for example, the idea is that that irreversibility may have something to do with how complications develop. And then why in diabetes are complications so hard to interrupt? So obviously we know, of course, there's many, many reasons, but we believed that one of the reasons could be the accumulation of these molecules that are highly enriched in hyperglycemia and oxidative stress, two conditions really that are very common in diabetes, diagnostic and also mechanistically, and therefore the bulk of the early research was to try to identify cell surface receptors. Once the RAGE was identified, we found that it was a very interesting molecule, actually a member of the Ig superfamily. You can see on the little cartoon here that it made its way into this family by virtue of a variable type domain and two C-type domains. Very short, a highly charged cytoplasmic domain, and really probably the most important discoveries about the RAGE really occurred after the identification of the AGEs. And that is because in addition to advanced glycation end products, molecules that now are considered damage-associated molecular patterns, such as S100 calgranulins and HMGB1, also can bind to RAGE and activate cellular signaling and mediate really chronic stress type conditions in the cells. It's very important to note that RAGE is expressed in all mammals, low levels in homeostasis except for the lung, where it has its highest endogenous expression, and that in the human and animal model system, in homeostasis in all of the other except lung organs, expression of the receptor is very low, but the ligands actually contribute through, at least in part, NF-kappa B binding elements in the RAGE promoter to upregulation of RAGE itself. So we sometimes would call it a feed-forward loop, the idea being that as the ligands accumulated in chronic disease such as diabetes, they would interact with the receptor, and one of the consequences was also upregulation of the receptor. So in terms of how to break the cycle, it's not a surprise that diabetic complications is a difficult system to try to interrupt. So after the discovery of the receptor, really one of the very first animal models that we had gone into was in diabetic atherosclerosis. And after the publication of the first paper, a researcher, Francesco Cipollone, contacted me because he had samples from carotid endarterectomy from humans. And we had just generated the antibodies, rabbits and guinea pig antibodies. And in fact, after a lot of vetting, the usual vetting, we shared those antibodies with Francesco, and he was the first to show actually that the RAGE was expressed to higher degrees in diabetic atherosclerosis versus control. He did this by immunostaining and also by Western blotting. And he also showed the very interesting finding that as the glycosylated hemoglobin level of the person rose, so too did the expression of RAGE. So when we saw this result, we were very excited because this is what we saw in the promoter luciferase studies, right? That the ligands could upregulate the receptor. And this would be the implication that the higher the glucose level, the higher the AGEs. So then after the discovery of the receptor, which took an enormous amount of protein chemistry work and all of the obvious vetting that one needs to do, we then wanted to ask mechanistically, even though these AGEs can bind to RAGE, does it have anything to do with the pathogenesis of atherosclerosis? So in the very first studies, we rendered ApoE knockout mice type 1 diabetic using streptozotocin, which is a beta cell toxin, which essentially reduces insulin levels in the animals. And pretty much the first thing that we saw for sure was that in the diabetic versus the control mice, there was clearly an increase in the atherosclerosis. We then decided, how are we going to try to interrupt the RAGE? This was long before the development of the knockout mice. And we were able to use a soluble form of the receptor, the decoy S-RAGE, which is the VC1C2 that I just showed you. We generated it in the lab in a baculovirus expression system, and we carefully characterized the T1/2, et cetera. And we administered it to mice compared to a vehicle. Vehicle was a protein vehicle. And in the very first studies we published, we were able to show that the soluble RAGE treatment reduced the mean atherosclerotic lesion area in the mice, and very, very importantly, without affecting the glucose levels and without affecting the cholesterol and triglycerides. So that was the first sense that what was going on here was that this RAGE pathway was a mechanism that was independent of glucose itself, but clearly from multiple follow-up studies, what we saw was that evidence of NF-kappa B activation and inflammation in the aortas of the S-RAGE treated mice was dramatically reduced. So one of the most wonderful things for me moving from Columbia to NYU was the ability to work with Dr. Ed Fisher. So Ed was also studying diabetes and athero, but Ed was studying it in a unique way. He was looking at regression of atherosclerosis. So this is a common comment when you're only looking at progression. You know, people will say, and rightly so, yeah, but what about reversing or regressing established disease? So this little cartoon here basically shows all of the work that we did together with Ed. And this particular model, so there's multiple models that they use of regression. This model is the transplant model. I particularly like this one because what I was interested in was the macrophage properties. And I knew that if we had a phenotype, we could use a CD45-1, 45-2 sort of strategy in order to try to figure out where the mechanism in the macrophages was coming from. So in these experiments, you render, these were LDL receptor knockout mice, diabetic. Again, we used streptozotocin and then fed the mice Western diet, high cholesterol, for 16 weeks. Then the arches are transplanted into also diabetic mice, but these mice are wild type. So basically what you're doing is you're taking those arches from a hyperlipidemic environment into normal lipid levels. And then the animals in our published paper, we sacrifice them at different ages and all the different controls, et cetera. But the most important thing of what we discovered was that compared to transplanting these arches from the diabetic atherosclerotic mice donors into wild type mice, the third bar shows that when we transplanted the arches into RAGE knockout mice, again with diabetes but normal lipids, there was an acceleration of the atherosclerosis. In the published paper, we show not just the H&E, but we show that the macrophage content greatly reduced, oxidative stress reduced, and also oil red O content, the lipids, were also greatly reduced, and more significantly so in the RAGE knockout mice. So as I mentioned, the reason why I was so intrigued with this model is we used the chimeric approach and then retrieved the CD45-1 versus the CD45-2 macrophages from the recipient aorta. And basically what we found was totally unexpected, multiple pathways that suggested that the deletion of RAGE affected interferon signaling and interferon stimulated genes. So the particular interferon that was the headliner in the RNA-seq was IRF7. And so IRF7 is something that we had never considered as being related to the RAGE pathology, but in fact, in our published paper, we were able to show that if we just looked at the entire macrophage content in the aortas of the recipient, wild type versus RAGE knockout, that the IRF7 expression was greatly reduced. Renu Vermani shared with us human diabetic atherosclerotic plaques, and she helped us to be able to show that IRF7 was highly expressed in the human diabetic atherosclerotic lesion macrophages. We also showed in our mice that the levels of plasma interferon gamma were actually lower in the plasma of the diabetic recipients that were devoid of RAGE compared with the wild type. So then we embarked on cell culture experiments to try to test the hypothesis that the ligands were part of the mechanism for upregulation of IRF7 and that it was working through the RAGE pathway. And so lo and behold, what we found is in bone marrow-derived macrophages, if we expose the macrophages to the damage-associated molecular pattern ligands, the upregulation of IRF7 was significantly attenuated. And then we wanted to look at, well, what are some of the effects in the macrophages of the IRF7? And so, as you would expect, many genes that were related to inflammation, shown on the right, were affected by IRF7 silencing in the macrophages exposed to the DAMPs. We saw that ARG1 and IL-10, which are more or less protective, were upregulated, and some of the more pro-inflammatory genes like TNF, NOS2, as examples, were significantly lower. But in our control panel, we also wanted to look at genes related to cholesterol metabolism, and we were very surprised to find that in the IRF silenced macrophages exposed to the DAMP serum, we found that there was upregulation of ABCA1 and G1. The two genes that encode LXR alpha and beta were also upregulated when IRF7 was silenced. CD36 was also lower. And some of the genes involved in cholesterol synthesis metabolism directly were also affected. So we haven't followed up yet on this with respect to the IRF7. One of the problems was, even though Dr. Tokiyama very generously shared with us the global IRF7 knockout mice, we quickly realized that for vascular biology, atherosclerosis studies, that there were competing interests of different cell types going on in the IRF7 global knockout mouse. And so we just finished and characterized the production of a floxed mouse, and I believe that this mouse will be much more helpful to us to really be able to figure out what the different cell types are doing. But suffice it to say that at this point, whereas we wouldn't have been too surprised about inflammation genes, we were very surprised that IRF7 may actually have these effects and on cholesterol metabolism. And in fact, there have been some recent papers that have linked this to pathways related to lipid metabolism. And so hopefully with the mouse model that we've generated, we may be able to understand this better. Obviously, the investigators who discovered IRF7 say that it is the master regulator of the type 1 interferon response. And all of their research, as you would expect, of course, is in viral infections. So this also opens up new areas for discovery of, like, why do we have RAGE, right? Like, what is the evolutionary reason for this? And we have no answer to that question, especially and particularly with respect to the viruses, but it's definitely an area of study. And I was very fortunate to just just begin collaborative studies at NYU with Kamal Khanna on infecting our RAGE-modified mice, and these ones also, obviously, with SARS-CoV-2. So we don't have any experiments yet, but that's certainly where the future will take us. So this slide just summarizes many years of research in diabetic complications. It's very clear that when we shared our soluble RAGE and also our knockout mice with many different people, they were able to link the receptor to, in animal models, mechanisms of diabetic kidney disease, neuropathy, and a very important result actually that we have done ourselves in our own lab is in wound healing. This was an important experiment because one could argue that a receptor playing such a role in innate immunity might disrupt wound healing. But in fact, if you apply the soluble RAGE to the wounds directly, the wound healing is actually accelerated. So we don't really know, and I'm curious about what the specific mechanism of that is. And one thing for sure is that with the advent of spatial transcriptomics, that may be a perfect environment to be able to figure this out. If this is to be a target for therapeutic intervention, we need to know the ins and outs of exactly what the antagonist might be doing. So as I mentioned, I really feel that if I look back on, what were the transformative events, I'm pretty sure that it was the idea that the HMGB1 and S100 calgranulin, also part of DAMP family, actually is probably the most important contribution here. And why are these? And what's so fascinating and what really, was very hard to understand when these molecules came off the sequencing, what are they? This is a non-histone binding protein, and S100 calgranulins are intracellular molecules that bind calcium, and they're involved in calcium-mediated signaling. So what we hypothesized, and now other researchers have proved, is that when cells are stressed, and obviously stresses come in many shapes and forms, depending upon the type of cell, that these types of molecules gain access to the extracellular space and either in an autocrine or paracrine mechanism are able to stimulate receptors such as RAGE, such as TLRs, and be able to really activate pathways that lead to chronic disease. So one of the questions that we were asked over the many years as well, you're studying complications of diabetes, you're talking about inflammation. Is there any link of this receptor directly to the development of obesity? And this was a very, very important question because it really begged the idea that, is it such a finite localization that this is only involved in complications of hyperglycemia and not maybe something more proximal? And so a paper was published which showed that in humans with obesity but without diabetes, that the AGE ligands, here called the carboxymethylysine, and in parallel, the expression of the receptor were both increased in humans with obesity without diabetes compared with lean humans who were otherwise very healthy and obviously had no diabetes. So the experiments that these investigators did in the human showed that the cells that were expressing the ligand and the receptor often overlapped, and that they were at least in part macrophages, vascular cells, and one of the first demonstrations of RAGE expression in adipocytes. So with Dr. Tony Ferrante at Columbia, we began studies in mice that were globally devoid of RAGE. And we were very surprised over multiple cohorts to show that the RAGE deficient mice, that's this blue line here with the circle shape, diamond shape, on the high-fat diet, this is a 60% high-fat diet, that these mice were pretty protected from diet-induced obesity. This occurred in the absence of differences in caloric intake measured in a chamber, and also just by measuring in versus out. And also there were no significant differences in physical activity in these mice. So one of the questions that arose, of course, was, well, are you sure that under any circumstances your RAGE-deficient mice could achieve obesity? We bred them into DB/DB background and showed that the RAGE-deficient DB/DB mice were equally obese to the controls and equally hyperglycemic. And in separate experiments, we used C57 Black 6 wild-type mice. We did not have any intervention except for the onset of high-fat feeding, and then 3 weeks after the high-fat feeding, and where you can see with the arrow that the animals were beginning to develop increases in body mass, we then began the soluble receptor. And essentially what we found was that despite no change in food intake or in physical activity, the animals that were treated with the soluble receptor were partially protected with significantly lower body weights compared with the controls. We looked at the fat because we really realized immediately that these were unexpected findings and that what is, explaining this. And in fact, when we looked at the fat, this particular slide shows the brown adipose tissue of the SRAGE treated animals, but the same is true in the animals that were globally devoid of RAGE, that uncoupling protein 1 expression was dramatically increased in the SRAGE treated mice compared with the vehicle. Once we saw this result, we sent the mice to one of the mouse metabolic phenotyping cores in the US, led by Jason Kim. And Jason showed that the knockout mice actually had a higher energy expenditure in the metabolic chamber. Importantly, Jason confirmed no difference in the food intake, so that this clearly was an endogenous mechanism that was stimulating this response. He also showed in the hyperinsulinemic euglycemic clamp that the RAGE deficient mice on the high-fat diet, in parallel with reduced body mass, were also protected from insulin resistance. So based on Jason's results showing that there was greater degree of protection in terms of energy expenditure, we had just generated and brought to NYU at that time our RAGE flux mice. So the gene is called AGER, which is why it appears as AGER, and the protein has retained the name RAGE. So in the first experiments, we deleted the receptor from the adipocytes. And basically in these figures, the green are the RAGE deficient adipocyte mice, and the red are the mice where the RAGE expression is retained. So these are the Cre negative, and the promoter is the adiponectin Cre recombinase mice. So you can see two things immediately, that the deleted mice, the AGER deleted in the adipocyte, gain less body mass than the controls, but very importantly, it is not identical to what we saw with the global knockout. And this is not a surprise. Obviously, there's probably a lot of cross-talking. We're trying to figure out what the other cell types are here, but for all practical purposes, what we saw and we see over and over again in our research is that the AGER pathway, RAGE pathway, does not affect in its deletion or antagonism lean mass in the mice, but rather it's the fat mass on 3 and 6 weeks of the high-fat diet that was significantly lower. Here, we also placed the animals in the environmental chamber, and we did indirect calorimetry to show that just like Jason had done with the global knockout, the adipocyte-specific knockout had a higher energy expenditure. So this is the insulin tolerance test. Now, this was done pretty much near the endpoint. Normally in our experiments, we keep the animals on the high-fat diet for about 12 weeks, and we do the ITT at week 10, and we do the glucose tolerance test at week 11. And here we did the ITT as stated, and we saw that they were remarkably protected. You can also see even at the time 0 that they're fasting on glucoses. Mice are fasted before the sacrifice for about 6 hours, were also significantly lower. The fat tissue, particularly the visceral adipose tissue. In the mouse, it would be called epididymal because these are the males, also showed a remarkable decrease in expression of inflammatory markers. What's very interesting, of course, is that, what is the hierarchy, which comes first, et cetera. So there's so much to learn. But one good thing is that, having the different mouse models and the human tissue can really help us go back and forth to try to figure it out. One of the first things that we did after the demonstration of the adipocyte-specific knockout is we thought about, well, is this effect coming directly from the adipose tissue? Because if you make an adipocyte-specific knockout, it doesn't mean that it's not coming really from some other depot, like brain or other. So in these experiments, we took the two different groups of the Cree mice. So the red, again, is the Cree negative. So there we have no deletion of RAGE. The green is the mice that are adipocyte ager deleted. And we fed them the high-fat diet for 12 weeks. And then we transplanted their fat into normal wild-type B6 males. After the males recovered from the surgery about 1 week, we then started those recipients on the high-fat diet. And basically, we were able to show, here you see in this experiment, the brown adipose tissue transplant really shows that, at least in part, the brown adipose tissue itself is the site of where some of the RAGE-dependent mechanism is coming from. The animals had higher energy expenditure, those that were the recipients of the knockout fat. And we also showed that if we looked at the endogenous brown adipose tissue and white adipose tissue, so not the donor fat, but the recipient fat, showed higher levels of the UCP1, especially in those that received the knockout. So one of the potential mechanisms, and we're working very hard on this now, is this really opens up what is a big area in obesity research right now, how adipocytes and macrophages, just to name a few, are able to crosstalk across different organs. And so in the current way of thinking, we're wondering whether or not the RAGE pathway has some effect on the cargo of the extracellular vesicles that are being emitted from and transmitted from the adipocytes, the adipose tissue. So we've begun some very early experiments to try to test that idea. And it would be one of the first times that, for example, in the cargo of those EVs, we would be looking at potential downstream targets of the RAGE pathway in terms of microRNAs, long non-coding, and then, of course, obviously the proteome, metabolites, et cetera. It's also important to note that, in addition, the transplantation of the knockout fat also rendered the recipient mice more insulin sensitive. So we could really reproduce much of what we showed in the overarching experiment in the whole mouse in the transplant. So I don't have time to go through all of it, but I just wanted to say that it was really important for us to figure out, like, well, what is the mechanism? So just showing the UCP1 is higher in the knockout, this is not a mechanism. And it took a while to figure out. And as usual, when you have to dig deep, it can be a lot harder, but that's good. That's a good scientific challenge. And essentially what we discovered in the end was that the RAGE pathway is blocking these adaptive pathways on thermogenesis because the RAGE ligands suppress phosphorylation of protein kinase A targets, such as hormone-sensitive lipase, P38 MAP kinase, and that by doing so, it suppresses lipolysis and all of the mechanisms in the mito that lead to upregulation, ultimately, of the UCP1. We also know that small molecule antagonists have the same effect, and this obviously is one of the potential for therapeutic opportunity. So people will say all the time, how can you talk about all these bad things? That is this really possible, right? That such a molecule would only exist for the purposes of, mediating chronic disease and chronic stress. So evolutionary biologists published a number of papers where they were looking at evolution of different molecules. And for whatever reason, they added rage to it. And what was amazing to see immediately is that rage is only present in mammals. And so we definitely don't have the answers. These are just phenomena, observations. But this particular group, the Laurustheatria group of mammals, these are the ones that are members of eutherian mammals. And these are the mammals that can use UCP1 as a measure to regulate their thermogenesis and heat. And so we hypothesized that perhaps in simpler evolution, when there was need for foraging for food, hunter-gatherers who rarely became obese, as we know, that this rage pathway may have been a mechanism to store fat, and in doing so, be able to then have that nutrition available if and when needed. However, in settings of chronic overnutrition, this pathway may be nevertheless activated, doing its thing, suppressing pKa, suppressing thermogenesis, and obviously this could be one of the contributing factors. I would note too that there's more that's interesting about this, and that is that if you look at the other receptors for DAMPs, the toll-like receptors obviously did not evolve initially and only in mammals. So how nature has set up the ability to handle nutrition, to handle inflammation, is really incredibly interesting to think about. And hopefully, researchers now and hopefully in the future will be able to unravel this. Why is there a rage? So again, it's always been the question, what are the homeostatic roles for the receptors? And this is what we can hypothesize, right now. Now, another major challenge that occurred over the years in the RAGE work was to understand how the RAGE signals. The cytoplasmic domain, not unlike other members of the Ig superfamily, does not have endogenous tyrosine kinase activity. We use the RAGE tail, we call it, the 43 amino acid just downstream of the single transmembrane domain in a yeast 2 hybrid assay, and were able to find binding molecules that could interact with the RAGE tail. And from that screen, there was only one molecule that came up. It was called diaphanous 1. And diaph1 actually is very interesting because it has a lot of parallels to RAGE, and it also opened up new ways that were critical for thinking. First of all, diaph1, just like RAGE, is expressed in many of the same cell types. It doesn't have that high level only at homeostasis in the lung that RAGE has, but diaph has multiple domains. It's actually the FH1 domain of diaph that binds to the RAGE tail, but it's a much larger molecule and obviously has other functions. So we were not surprised when we read that diaph was related to Rho-GTPase signaling. Many labs have shown that RAC1, CDC42, even Rho-A can be activated by RAGE ligands through RAGE. We knew that RAGE, and now diaph does the same, is involved in regulation of gene expression through serum response factors. And very importantly, when we obtained global diaph1 knockout mice, we rendered them diabetic and found that they were protected from many of the complications that the RAGE knockout mice were. And very importantly, they also had the phenotype that when they were protective, there was no difference in the levels of glucose. So the diaph deleted animals were very similar to the RAGE deleted mice in the diabetic complication setting. So one of the wonderful outcomes in team science, which is what we try to do every single day really, is to bring new collaborators, to bring new insights and stories. And so my colleague Alex Sheckman at the University of Albany was very excited to work on diaph. He found the extracellular RAGE to be way too complicated, too many domains, too many ligands. He loved the idea that this tail was very short and really was something that for an NMR spectroscopist he could work with. So Alex was able to show totally independently that RAGE tail bound to DIF1. He identified the amino acids in the figure. The model is the RAGE tail. He showed that R5Q6 and the RAGE tail were critical to interact with DIF1. He mutated them and showed that the binding was reduced. We used the same constructs in cells and were able to show that in the alanine mutants, the RAGE ligands no longer activated signaling. So essentially, there are probably other molecules that can bind to the RAGE tail with, outcome such as signal transduction differences, but at least we can say for now that multiple different experiments, including using mutations, show that the RAGE tail and interaction with DIF1 is important for RAGE signaling. The full scope of this is obviously yet to be detailed, but is obviously a very important work in progress. So the shock that came from some of our experiment, which proved to us that DIF1 transduces the effects of the RAGE ligands, but more than that. So we wanted to understand how DIF1 was related potentially to atherosclerosis. And the reason this was so important is that obviously in the case of trying to move forward with small molecule inhibitors, if there were an adverse effect of deleting DIF1 in athero, this would not be good because this obviously is very important, cause of changing health span and lifespan in people that live with type 1 and type 2 diabetes. So in our very first experiments, what we did is we rendered global DIF1 knockout mice, type 1 diabetic, again with streptozotocin, and we found very similar results to what we found in the RAGE. We found that if we looked on FOS, shown on the left, and if we looked at the H&E, we saw that the mean atherosclerotic lesion area was remarkably lower in the double knockout, that's LDL receptor knockout and DIF knockout, compared to the controls. These mice were litter mates, and very importantly, the serum concentrations of glucose did not differ. So it was very similar to what we had seen before. Now, what was very surprising, and we didn't really expect this because we never really saw this so blatantly in the case of RAGE, is that the plasma concentrations of cholesterol and triglyceride were significantly lower in the double knockout. With Dr. Fisher, he helped us to do the FPLC study shown in the bottom panel. And basically, it's what you would really expect, obviously, for LDL receptor knockout mice, right? There were no differences in the pattern of the HDL, but that most of the differences were that the double knockout had lower VLDL, IDL, and LDL. And we also looked together with Dr. Fisher at the livers of the mice, and we were able to show that the oil red O, using as one marker for lipid content, was lower in the lipids of the double knockout mice. It's also important to state that we looked with biochemical assays with and without cholesterol esterase, and in both cases, and in the case of the biochemical measurement of the triglyceride, that these were lower in the double knockout compared with the single knockout mice. So we had a long meeting with Dr. Fisher to go through, first of all, this was unexpected, but that's wonderful, right? How this DIF is regulating lipid metabolism. And I do believe we're really only scratched the surface. This is a very complicated story, but I'll show you what we were able to publish as first pass to at least be able to get the result out there. And, we're trying to use all of the knowledge that all of us have with respect to lipid metabolism and also what DIF does to try to put the pieces together. And so Ed suggested, he said, let's just start with bulk RNA sequencing. So the bulk RNA sequencing, not too surprisingly, showed what we would have expected, and that is that genes that were directly related to hepatic lipid metabolism were significantly lower in the bulk RNA-seq, but we also did a full panel of experiments where we validated the results using real-time PCR. And so what you see here, so this was the replication of the primary findings in the bulk RNA-seq. So you see lipin-1, lipin-2, lower. ACA, CA, and B, lower. GPAT2, lower in the double knockout, and FASN, lower. And so these then were the pathways that were not significantly different. So the first thing we thought was that SREBF1 and BF2, that these would be different. They were not. And it was verified in the livers using the real-time PCR, so we were sure of this result. Here we saw that there were no differences in RXR alpha, the two genes that encode for LXR alpha and beta. We saw no differences in the PPARs, and we saw no significant differences, and for sure the bulk RNA-seq showed no differences in the efflux molecules. So this got very complicated very quickly because some of the obvious pathways, we just didn't see differences, and science is what it is. So then we took the livers and we wanted to look at the levels of the proteins of SREBP1, BP2, and also CHREBP. So even though we did not see differences in the mRNA for these molecules, we also took the whole liver and also looked and showed no difference in total protein for SREBP1, 2, and CHREBP. But what we found was that the nuclear content of these molecules was significantly lower in the double knockout versus the single. So this then created a situation where we'll go right to the literature, and lo and behold, there was a very interesting body of work on regulation of molecules that did not directly affect mRNA for SREBP1 and 2, but actually affected downstream pathways. And so we wanted to look in these animals because we started looking at AKT pathway, mTOR pathway, multiple pathways that could have explained our result by virtue of a metabolic difference. And in our published paper, we saw none of it. We were not able to see that the deletion of DIAF affected any of those pathways. Strikingly, just continuing to probe and go down what the possibilities were, we were very surprised to find the result on this particular slide. So the ROC1 pathway through LIMK1 is a mechanism for controlling phosphorylation of cofalin-1. And lo and behold, what we found was that in the livers of our double knockout mice, phosphorylated cofalin, that's the serine 3A, was significantly higher. I would also say that the first thing we did is we took aortas and other organs and found that phosphocofalin was much higher in the double knockout. And it's really important to note that cofalin is a very complicated molecule, but something that really intrigues me right now to go after what this is, because other people had shown that cofalin-1 could control the nuclear translocation of SREBP1. They never showed a mechanism, they never showed how cofalin did that, but they showed that relationship. I also want to point out that the serine 3 phosphorylation of cofalin inactivates it. So what the DIAF deletion is doing is rendering cofalin inactive. And why is cofalin so interesting? Well, cofalin turns out to be directly related to actin polymerization and mechanisms by which the cells can either sever formed actin to create the products to be able to lead to new polymerization. And so it really has a dual role. But how this cofalin can be regulating SREBP1 translocation to nucleus is not described at a mechanistic level, but it's a beautiful scientific opportunity. So because of knowing that there was this relationship with actin and actin polymerization, we began working in a cultured line, HEPA-1-6 cells, which are hepatocyte-like cells, not perfect, but they're mouse. And so what we found is that, not surprisingly, right, that silencing of DIF-1 actually reduced F-actin intensity in these hepatocyte-like cells. We also showed in the hepatocyte-like cells that if we depleted cholesterol, we used HPCD to do that, that if we silenced DIF-1, we could significantly reduce the nuclear translocation of SREBP1, not of CHREBP. So this then was just circumstantial evidence, right, that this could somehow relate DIF and nuclear translocation of SREBP1 and maybe actin to the whole picture. So the next experiment we did is we did the same thing in our cells, but we treated them with latrunculin B. And so this molecule is a small molecule that blocks actin polymerization. And lo and behold, in our cholesterol depletion model in the cells, when we treated the cells with the inhibitor of actin polymerization with the stimulus of reducing the cholesterol, we were able to reduce the nuclear SREBP1. So the next experiments now hinge entirely on trying to put these pieces together. We have done some early studies silencing cofalin-1 in the hepatocytes, and we can show a direct relationship to the silencing and the nuclear translocation. And the colleague I mentioned to you before, Alex Sheckman, has discovered that cofalin binds to DIF-1. This had never been identified before. So we believe and hypothesize that one of the mechanisms by which DIF-1 can be contributing to regulation of lipid metabolism is through cofalin/actin cytoskeleton dependent mechanisms. There's so much to do in this, and we just generated the cofalin-1 flox mouse because there's no way to do these experiments. First of all, cofalin-1 global deletion is embryonic lethal, failure of fusion of neural tubes in the global knockout. So we hope that now we have a new research tool to be able to try to sort this out. But it's so fascinating, right, that, just desperately testing phenocopying opened up an entirely new aspect of DIF-1 biology. So finally, it is pretty well established that the RAGE extracellular domains are really very promiscuous. Ligands can bind at all those different domains. The same ligand can bind to more than one domain. So in terms of trying to develop a therapeutic, Alex and I hypothesized that if we focused on the RAGE tail binding to DIF-1, that we might have the beginning of a novel form of therapeutic intervention. So Alex and I were funded from the JDRF to screen a 59,000 compound library. We wound up with 13 hits. NYU brought in a medicinal chemist that found tremendous similarities through multiple years of blood, sweat, and tears with this structure-activity relationship and ADME PK and all of these things, Ames test, the kind of things that academics like us who do signaling would never even know what they meant. We had to quickly learn what those things were. And with wonderful colleagues that helped us, we actually published just a few years ago about a novel chemical probe. It's not a drug. It can never be a drug. But it was a novel chemical probe that was a huge advancement over the molecules that came off the initial screen. And so between our lab, we do all the biology and all the in vivo, and Alex's lab does all the binding. This is the fluorescence binding assay. Alex also made a FRET assay for this. And so the bottom line is that, at least in terms of the affinity, these molecules were much better after the medicinal chemist did all of that hard work with structure, activity, relationship, refinement. But the bottom line is, what about in the mouse model? So this is work that we did in type 2 diabetic mice, where we fed the mice for 4 months in a medicated chow with this chemical probe RAGE 229 versus vehicle chow. And with our colleague at Columbia, Dr. Dagati, we were able to show very significant benefits on multiple indices of the pathology, sclerosis, podocyte effacement, and also thickening of the glomerular basement membrane. This was true in male mice and in female mice. And then in our lab, we used the metabolic chambers to basically examine urinary albumin excretion over 24 hours. And in both male and female mice, in parallel with the pathology endpoints, we were able to show that the albumin excretion was reduced. And very importantly, it brings me all the way back to 1998 when we did the experiments in the ApoE knockout. These animals had no difference in their glucose levels. So it was occurring at a more disease-modifying activity directly in the kidney. Now, one thing I can tell you is that there are influences of immune cells that infiltrate the diabetic kidney, and we're trying to work out those mechanisms using the flox mice for RAGE and for DIAF and on myeloid specific promoters. So nature has created its own biomarker, which may be very helpful in the future. It's called soluble RAGE. Total soluble RAGE is a combination of a cleaved form of sRAGE from the cell surface and of a splice variant that is located in the C2 domain that essentially removes the soluble component right off of the cell surface. The soluble RAGE has been shown by many labs now, in humans actually, to be mutable by therapies. So people that are treated with statins, with ACE inhibitors, these types of things, you see pre and post that the sRAGE levels rise after the treatment. And the hypothesis is that the rising of the sRAGE means that there's more material available to decoy and trap these ligands before they can bind to and activate the cell surface receptor. But it's not that simple. As usual with this molecule, there's just a lot of other issues. And what started to show up in the literature were people who were showing that low levels look to be helpful to the human, but high levels in other settings appear to be helpful. And so in order to try to address this, Dr. Rauchi just published on her work, where she suggested that looking at the ligand and the receptor in a ratio is actually more valid and more logical if you think about the science than just looking at the sRAGE alone. And in this published paper, she did a retrospective study where she showed that the ratio of the ages to the soluble RAGE were associated much more significantly to all-cause mortality and cardiovascular complications in people with type 2 diabetes versus only looking at the age levels or only looking at the sRAGE levels. So this may actually be, hopefully for clinical trials, going forward, if we ever get to that point, this may be an important biomarker, not only measuring measures of inflammation, right, in plasma in humans, but perhaps also looking at age sRAGE ratios. So in closing, I want to acknowledge the current and past members of the lab. I mentioned most of the collaborators directly involved in this work. And also in closing, I just want to say, on behalf of all of us in the scientific, I look at my research now, and then I look at everything that Bob Eckel, and how many times I had to, let's search Bob Eckel, let's read what he would say about this. And, it's just so wonderful that you all have this wonderful opportunity here, that you've been really so fortunate to have worked with Bob for so many years. And even those of us who didn't directly work in Denver, for 40 years, are recipients of all of his leadership in diabetes, metabolism, lipid research. He's just known as a tremendous mentor and has a tremendously generous colleague and friend to so many of us. in the scientific community. So I applaud you for this wonderful opportunity to, really acknowledge him. And it's just been a great pleasure spending the morning with Bob and his wonderful wife, Margaret. And I thank you all for the opportunity to come here and to help honor Bob. Thank you.
And we're just about out of time. I think we have time for one or two quick questions. I'll throw this out to the audience. Let me find my way back here with the microphone.
Really fascinating work. My question relates to the connection between RAGE expression and IRF7 expression in the macrophages. Was that specific to IRF7, or did you see other IRFs, IRF3, IRF5 also were regulated by RAGE?
Three, right? Only three. So we looked at the others and that was the only one. And it makes sense, right? Because they're kind of like in partnership, those two, right? So the effect wasn't as dramatic and it didn't show up in the RNA-seq, interestingly, but you can miss a lot, right? But yeah, there was a significant attenuation in the RAGE knockout macrophages, just of three.
One other question?
Thank you very much. That was a tremendous talk. I was wondering, you saw in your S-RAGE treated mice increase in energy expenditure. UCP1 generates heat rather than ATP. So did you look at core body temperature or local temperature of the fat depots at all?
Oh, of the mice? Yes. So in homeostasis, it's identical. We also put the mice, we had a colleague that had a thermoneutral chamber, and even at thermoneutrality, there was no difference in their core body temperature. But when you stress them and put them in those same chambers at 4 degrees, the global RAGE knockout mice can protect their body temperature. They have significant higher rectal temps compared to the controls. So it's all part of that same overarching mechanism of conserving your thermogenesis.
We're right at 1 o'clock. So I just want to say, Dr. Schmidt, thank you very much for being here.
Thank you. Thank you.